I have the privilege today of introducing Gordon Burghardt, who has to stand next to me not because we're in a picture, although we are, but also because you're supposed to be able to hear me better because he has wearing the microphone. So that's what this is about. Gordon got his PhD and actually his BA or BS and his PhD from the University of Chicago. He then went to the University of Tennessee, where he's been ever since with a few visits and things. He is there the Alumni Distinguished Service Professor, Professor of Ecology and Evolutionary Biology, and Professor of Psychology. He is a fellow of as many organizations as you care to list: AAAS, APA, APS, ABS. He's been on, if you can believe this, even more editorial boards. I'm not sure that I have enough time to read them off.
Don't.
But among them, Journal of Applied Animal Welfare, Journal of Comparative Psychology, Developmental Psychobiology, Journal of Chemical Ecology, Journal of Herpetology, Zoobiology. He's been the associate editor of AL&B and the American editor of Ethology. He's edited at least 4 books and written another one, I think, which is in the process of coming out on play. He's specialized in things ranging from iguanas to snakes, which may not seem like a large range from our perception, but is a large range within biology. He's published so many I couldn't count them. Now, he's published more than 100 chapters. He's written more than 100 journal articles, more than 50 chapters, and about 30 reviews. He is specialized over the year in a number of different things. His interests range from black bears and all of the issues related to them as they appear in the mountains around Tennessee. He's done a lot of work on iguanas, done a lot of work on play, he's done a lot of work on ethical aspects of dealing with animals, and is going to talk to us today probably about the many experiments he has done on relating to snakes. Is that true?
A little, only a little.
So it's a pleasure for me to introduce to you Gordon Burghardt.
I'm very pleased to be here to talk to you about some aspects of evolution and behavioral plasticity and behavioral variability, trying to put several things together. And what I want to start with, of course, is to mention that Charles Darwin, who we see as one of the founders of modern biology, also had a very great interest in psychology. And if you look at his notebooks prior to the time of the origin of species, they are filled with questions and comments and queries about everything from how the brain works to human behavior to ethics, cultural variations in behavior, mating behavior, human behaviors of all kinds. However, little of this, in fact, none of this, appeared in The Origin of Species, his seminal book in 1859. However, at the very end of that book, after 500 pages of barnacles and cats and dogs and pigeons and honeybees, he had this intriguing paragraph: "In the distant future, I see open fields for far more important researches. Psychology will be based on a new foundation, that of the necessary acquirement of each mental power and capacity by gradation. Light will be thrown on the origin of man and his history." And shortly after Darwin wrote, in the late 19th century, there was a flowering of interest in animal behavior, comparative psychology. It was compromised, as we all know, by emphasis on anecdotal evidence, on anthropomorphism, and other problems, and became dormant in psychology, particularly American psychology by and large. It did get a resurgence in Europe, primarily in ethology. And ethology I define as the naturalistic study of behavior from an evolutionary perspective. And one of the founders of modern ethology was Nico Tinbergen, who received a Nobel Prize in 1973. And a few years earlier than that, he wrote a seminal paper on the aims and methods of ethology. And he came up with 4 basic aims that we need to focus our attention to in understanding any and all behaviors. It was a holistic approach but he tried to parcel out the kinds of questions that we need to answer. And he claimed that we needed to have answers on all these before we can claim to have any competent understanding of an animal's behavior. Now, the first one, which I call control, is one that should be very congenial to this group. He called it causation. Sometimes we talk about the mechanism of behavior, proximate factors, and so on. But we're trying to identify the internal and external factors underlying behavioral performance. The second one, the issue of function. What is the behavior good for? Why does the animal do it? Adaptiveness, survival value. We've tried to identify the contributions of the behavior patterns that we see to individual, reproductive, and inclusive fitness. I will not go into those technical terms. Basically, we're talking about reproductive fitness in terms of the passing on of your genetic characteristics to the following generation. The issue of ontogeny is a very important one to ethologists. It's identify the patterns and processes in behavioral change during individual lifetimes. This is another one that actually has been the focus of more behavior analytic work with humans, I believe, in terms of infant and child behavior, than we usually find in non-human animal studies and behavior analysis. Of course, the hallmark one, then, is the issue of evolution, which question of phylogeny, and I also include here genetic and cultural inheritance. I think we need to consider both of these together, and in fact, there's been some very interesting seminal writings recently in the last few years looking for parallels between genetic and cultural inheritance. What you're trying to do here is to identify historical patterns and processes in behavioral change across generations and taxa. Now, most of us are aware of ethologists using behavior patterns to try to compare different species and see how different behavior patterns may have evolved, or using behavioral patterns to reconstruct the probable evolutionary history of a group of species. However, it's also important to look at change across generations. And this is termed microevolution, and that is, in fact, going to be the focus of what I'm going to emphasize in my remarks today. Now, the last one, after this morning, I feel I can more safely bring this up, the issue of private experience. I think we have to add a fifth aim to Tinbergen's. Tinbergen was very wary of subjectivism. He was still fighting the battles of vitalism and intellecties and so on. But private experience, called descriptive mentalism, as Nick Thompson has, felt experience, private events, Skinner's term. Basically, here we're interested in the patterns and processes in life as experience, and that may be very important. Now, just to give a little justification for the private experience role, I can do no better, perhaps, than quote my introducer, Bill Timberlake, who in an article in the Behavior Analyst several years ago wrote this, which I think is a very insightful and important comments. Experimenters not only need to put themselves in the subject's shoes, they need to wear them, walk, watch, hear, touch, act, I would say smell like the subject. The humility required to assume this role, coupled with the power of the experimental approach, should increase the efficiency with which the understanding, prediction, and control of behavior can advance. So I think the time is becoming ripe that we can introduce a fifth aim that we need to really understand, comprehend, if we want to say that we have a good handle on all aspects of behavior. Now, in recent years, there's been various approaches to using evolution in psychology. Darwin can be read in many ways by many people from different perspectives. And one of the ones that is very popular these days is evolutionary psychology, which has sometimes a very narrow focus, which I characterized here as a historical approach. It's based on the idea that there are ancient evolutionary processes that still inform our behavior in psychology in the present, but these evolved in a time, in an environment different from where we live now, before agriculture, before written language, before a lot of technology, before we lived in large social groups and cities and so on. And they characterize this as the EEA, the environment to evolutionary adaptiveness, and that is where our psychology was formed. Now, we have a mismatch between the environment in which we find ourselves and this environment in which our basic psychological and behavioral properties were shaped evolutionarily, and many of our social problems, many of our problems in society, from obesity to social interactions and intolerance and so on, are based on the differences between the EEA and our current environment. They focus here on species characteristic or gender-specific psychological and behavioral mechanisms. I'll tell you why they focus on gender ones in a moment. The interest here is in human universals, the fact that there's a human nature that we all share, and that is the key that we want to understand. Individual differences, by this perspective, are viewed as noise or largely environmental. So they would be very sympathetic to a more simplistic behavior analysis that only focuses on all people, all animals being the same, except for the contingencies of reinforcement that they experience during their lifetimes. This is called the jukebox theory. Not my term. A more complicated, but perhaps more useful approach in many respects is to consider that the processes of selection and adaptation are still occurring in the recent past as well as currently, and that we need to focus on individual differences a great deal, on individual other within species levels of selection, selection involving the individual, the family, the group, the society. In other words, there are levels of selection at which evolutionary processes can be acting, and we have to look at the fine-tuning of behavioral adaptations to current conditions, and hence individual differences are viewed as a dynamic interaction or product of genetic diversity and environmental conditions. Now, going back to Darwin, if we want to get a handle on the role of selection in behavior, which is the primary focus I'm going to be talking about, what did Darwin actually say? Darwin, remember, is not known just for his theory of evolution, because evolution was out there before Darwin wrote. It is because of his coming up with a mechanism, which he called natural selection, along with Wallace, in which there are several components. Animals have inherited variations. These variations lead to animals that are more plentiful than can possibly survive, and those animals with certain variations, genetically based or inherited variations, are going to survive, and then process of adaptation will occur. And survival selection is the main kind of selection that many of us are familiar with from the textbooks, process of adaptation to the physical and social environment. And a prototypic example is the shorter extremities and heavier fur in Arctic mammals. Let's compare those to those that live in the same species or other species living farther south. However, later in his life, Darwin developed this ideas of sexual selection. And he focused largely on animal behavior in these writings and on human behavior as well. And sexual selection is a concept that was not really readily understood by his contemporaries. In fact, it received relatively little attention by evolutionary biology until the last 20 or 30 years. And sexual selection, Darwin divided into two types, and these descriptions are pretty much used today in the same way that Darwin formulated them 120-some years ago. Selection of inherited characteristics, intrasexual selection, which is the selection of inherited characteristics that increase the chances of outcompeting others of your own sex for access to the most reproductively valuable members of this opposite sex. And male-male fighting is the example that Darwin used. It's still the focus of lots of research. We also now know that there's much female-female competition in animals and in humans for access to the opposite sex. However, it's not quite as violent or easily seen. It's more subtle, and so that's one of the reasons why it has not been focused on until fairly recently. Intersexual selection is, Darwin called it aesthetic selection. Selection of inherited characteristics that increase the attractiveness to the opposite sex. And here the prototypic example is the peacock's tail. This emphasis on sexual selection has driven much of the modern evolutionary psychology, which is why they have focused so much on gender differences and sexual differences, because it fits so nicely with this. And the evolution of sex is still, in biology, a conundrum. There's still lots of things we don't know why and how it has evolved. And if you look at the animal behavior, the journal on behavior, it is filled with articles on sexual selection more than probably any other aspect of behavior. That was not true 20 years ago. Okay, it has really taken off as something of great interest. I'm going to go back now a little bit to late 19th century and develop a different trend, or another trend, in looking at the role of selection, and building on the idea that selection can operate on species characteristics and, quote, create instincts or innate behavior patterns, but it can also operate on the level of the individual to change behavioral repertoires within the individual. And the first person that I know of who really put this forward in a very nice way, which has been virtually forgotten, was E.L. Thorndike, 1899. And I think this is worth reading because it has some certain very modern flavor to it. If our view of the process of association is correct, it is a process of selection among reactions, not by eliminating the animal that does not react suitably and so developing a stock with certain instincts, but by eliminating the unsuitable reaction directly by discomfort and also by positively selecting the suitable one by pleasure, and so developing certain associations in the individual. It is then selection within the individual that is the great case of plasticity, and is of tremendous usefulness in that it definitely enables the animal to modify his acts and so meet varieties and modifications of environment. New feeding, new foods, new friends, new enemies are dealt with by virtue of it. He who learns and runs away will live to learn another day. Now, this idea was, as many of you know, the heart of Skinner's Phylogeny and Ontogeny paper that came out many, many decades later, with this basic idea. And because of the virtue of this idea, this paper was considered so important, it was put in the canonical papers of B.F. Skinner. But it is not a new idea. Now, let us compare natural or phylogenic selection as Skinner called it, and ontogenic selection, which he called the behavioral one. The genotype is, of course, the genetic constitution of the animal. We have genes, and they have different alleles. And I may use the term allele, and that, of course, is the form of a particular locus on the genome. And we remember the big A's and little a's. We now know that you can have a whole alphabet. There can be 16, 20, 30 more alleles at the same locus. So, natural selection, which includes sexual selection or survival selection, depends on heritable genotypic variation and selection on phenotypes in a population. Ontogenic selection focuses on selection of behavior, which is the phenotypic character. So it depends on individual performing variable responses, called behavior, and selection on such variation by reinforcement contingencies. But is there anything equivalent to the genotype in this model? And mind or brain, both of those will be suspect labels. But should there be? Will eventually there be? And I think even Skinner at the end of his life realized that the brain was going to give us many of the secrets here. So in some sense, something has to be producing the variability, the source of the operants, that gives us the behavior that then can be selected. Now we can start off by making some few comparisons here between this evolutionary and behavior analysis approaches. First of all, it's interesting that the initial insights in both of these types of selection were based on domesticated species. Darwin relied on what he called artificial selection, what farmers and people have been doing for years in creating pigeons that did all kinds of weird things, dog breeds, and so on. And similarly, of course, much early comparative psychology and animal learning was based on use of domestic species. Both search for applications in the natural world. they're not just interested in the domesticated or artificial context or species. They want to have some power, some control, some understanding of what happens in the real world. We both rely on considerable variation either among individuals in a population or in behavior emitted by individuals. I think that's a very important comparison. There are conceptual similarities then in the analysis of populations of organisms or populations and behavior shown by individuals. Bill Baum, in an earlier lecture in this series, made that exact point. Populations of individuals or responses, not individual organisms or responses, are the key to the analysis of both pattern and process in both fields. Okay. You're dealing with probabilities, you're dealing with large numbers. And we also find that in both fields there are constraints that limit the power of selection. You can't move any animal to perform any behavior pattern. There are limits in terms of natural selection and artificial selection. There are constraints that one hits. One can only select in certain ways to certain degrees, opposing forces. Similarly, when the Brelands came out with their misbehavior of organisms paper, it made people sit up and realize that we can't do everything with behavioral contingencies without being a little bit more sophisticated in our knowledge of the natural behavior of the organisms and the various physiological and other behavioral aspects of their behavior. So, what can a geneticist tell us about behavior that might be of use to behavior analysis? Skinner and others who have made this analogy between natural selection and ontogenic selection or behavioral selection have never, as far as I know, really looked at what the geneticists and the population biologists do in their models to see if there's any ways that they may give us some insights into how we can look at individual behavior or altering behavior. Let me then go through three of the major kinds of selection that you find. All these illustrations, by the way, are standard ones you find in evolutionary textbooks. The first one is called stabilizing selection. You consider throughout normal distribution of behavior pattern on some variable. You can also think about this like the range of heights, for instance, or body weights, or mating speed, or any characteristic, behavioral or otherwise, that we are interested in. And in this case, the red are the animals that are selected. The ones that are either in the laboratory or in nature are the ones that breed and have offspring in the next generation. The ones in the extremes are eliminated. And what happens then is you reduce the variance in the subsequent generation. You increase the slope of the peak. You've eliminated, in other words, the ends. And this is something that many behavior analysts, many people are interested in accomplishing. Right? You want to eliminate people, classrooms, for instance, that are disruptive. The people are maybe too passive, the students who are too passive, those who are too disruptive. Conformity. This, in a way, is selection for conformity. Now, speaking of disruption, that leads to the second kind of selection, disruptive selection, in which it's the individuals that are favored are those that reproduce, are those at the extremes, and the middle, the animals, are not favored. So we have selection against the immoderate, the in-between. And this leads to increased variance, and actually may even lead to two separate curves around, normal curves around each of the extremes. And it is thought that by this process, new species may be formed, or within a population, within a species, different morphs can be formed. There are some snakes, for instance, in California, that have stripes. Others have bands. Exact same species. You might even find both forms in the same clutch of eggs. But you don't find checkerboard ones or ones that are sort of crisscross, right? It's one or the other. Somehow those that were in between clearly are selected against. The kind of selection that has been the focus of most animal breeding and the kinds of change that we may be interested in, particularly with human behavior, is directional selection. That is moving the mean, not changing the variance, but moving the mean. So only the ones in the top half of the distribution would be selected, and after several generations, the mean would shift. How does this happen? Can we, in fact, come up with ways of predicting in advance what role selection, how strong selection will be on subsequent generations? To do this, we have to look at some of the standard formulas that the geneticists have developed. Top main thing here is that the phenotypic variance is a product then, or additive combination of the genetic variance, the environmental variance, and the interaction variance. And if you look at the way most geneticists treat their data, you will find that they virtually ignore the interaction variance. I think that that's a serious mistake. There are some reasons why it has been ignored, methodologically, methodological being a large part of it. But I think that that is where a secret of a lot of future advances in genetics and environmental interactions and understanding behavior are going to take place. But focus just on the bottom here. If you have know the genetic variance and the total phenotypic variance, you then get a value called the heritability in the broad sense. Now, heritability is a concept that is often somewhat controversial, and it's usually the more controversial, the less you know about it. So hopefully we will know a lot more about it after today. What are some of the properties of heritability? First of all, it varies from 0 to 1. Okay, nothing controversial about that. The second point is one that is often misunderstood. It does not indicate genetic fixity or plasticity. Okay, everybody hold up their hands or look at their hands, and how many digits do you have? 5. Okay, I would dare say, does anybody here not have 5 digits? Okay. That being the case, if you calculated the heritability, it would be what? 0. Okay. Yet, no one is going to deny that the fact that we have 5 digits is largely determined by our genetic heritage. Okay. Similarly, you may have high heritabilities and yet be dealing with a very plastic trait, a trait that is very open to change by consequences, environmental events, and so on. Third point is population specific. You don't calculate a heritability once and have it forever. It can only be really used in a population in which you are focusing on. You can't generalize from males to females. We work with snakes, and we look at behavior of newborn snakes at birth to chemical cues from prey, to anti-predator behaviors, and we find that snakes from Michigan and snakes from population of Wisconsin differ in not only the mean traits in many respects, but also the level of heritability of those traits. So even within a species fairly close together, you can find quite considerable differences in heritability, which can have important consequences for how evolutionary processes can work. Fourth is that in behavior in particular, your measurements may often be imprecise. If you're dealing with number of scale rows on a snake or number of ventral scales, you can count them several times and get pretty reliable. Behavioral measures may not be as reliable. And so there's been the development of a measure called repeatability that I'll mention later. Now, most of the genetic calculations use standard kinds of linear models, and types of things you'd want to calculate would be the means, your variance on the trait, the covariance if you're dealing with several traits, standard deviations, correlations, and regressions. The genetic variability itself can be broken down into at least 3 categories: the additive genetic variance, dominant genetic variance, and epistasis or gene-gene interaction. The latter is generally found to be less than 1% of the total genetic variance and is often ignored. Additive and dominant variants are more common and many of the methods for determining heritability have a confound of these two. Why is this important? Because the additive genetic variance is the important measure for how natural selection can operate. And Fisher's R.A. Fisher, who we know from statistics, but also was a leading geneticist and evolutionist, his fundamental theorem of natural selection is that the additive genetic variance, that measure, gives us a handle on what will happen under natural selection. It's only the additive genetic variance. So this leads to the concept of the narrow sense heritability, which is the additive genetic variance over the phenotypic variance. And this is the important thing that we've tried to measure in trying to predict how a population will respond when selection occurs. Environmental variability is also broken down a little bit more, and I'm not going to talk about this in great detail here. We can talk about common prenatal and postnatal environments, and that environmental variation, which is due to non-genetic, non-shared influences. Assessing this in human populations can be rather difficult. One of the things that I like to use my snakes for, and people have used rats and mice and others, is that you have large litters of animals. And those animals you can, say, have a common prenatal environment. And then, of course, you can raise them with different parents or keep them in the same parents, raise them singly. You can then alter and control what aspects of the environment are common or not. More recently, there's been a lot of interest in maternal effects. That's another topic that we've been working on, and I won't have time to go into it in any detail today, except that in addition to all these other things we've been talking about, the phenotype of the offspring is also influenced by indirect genetic maternal effects and environmental maternal effects. For instance, it was the mother from a genetic strain that has large offspring. Then her offspring, if they are related to the mother's size, might be larger because of that genetic factor of the mother. Or the mother may be large because she happened to be in an environment where there was lots of food, okay, irrespective of her particular genotype. And one can try to parcel out those effects. So given all that, how do we go about measuring heritability? Not an easy thing in many cases. We can compare siblings. We can compare half-siblings. Same mom, different dads, or different dads with different moms. This gives us half-siblings, and this allows us to make a little better estimate of distinguishing between the dominance and the additive variance. We can compare parents and offspring, and that is the method that I'm going to walk through with an example later today. Monozygotic and dizygotic twins, the use of fraternal and identical twins, is the hallmark of many of the studies that we all know about the heritability of religious preference or buying lucky strikes or various remarkable things that have been shown for twins that were separated at birth and still have certain similarities. A very interesting way of measuring heritability is to evaluate the response to selection. And I will go through that a little bit too. Note, repeatability set the upper bound to the heritability estimates. So your heritability estimates can never be higher than the reliability of your measurements. And secondly, the narrow sense heritability is always going to be less than the broad sense heritability. Here, mean points of parents. So the shaded part on the graph on the right represents the midpoint, the mean of the midpoints of parents that are selected, okay, at the top end of the distribution. And you can see then that the offspring after one generation are going to, their mean's going to have moved over, another generation's going to move over a little bit more. How can we predict this? Well, we need to have two concepts. Forget the top one here. We'll just talk about selection differential, which is the mean of the population of selected parents compared to the general population. And secondly is the response to selection. That is the difference in the mean phenotypic value between the offspring of the selected parents and the parental population before selection. So the response to selection turns out to be equal to the heritability times the selection differential. And thus heritability is the response to selection divided by the selection differential. So if you then look at here's a normal distribution, you take parents that are 2 standard deviations above the mean. If the heritability of that response is 0, then no matter what you're selecting for, you're still going to end up with no change in the mean. It's going to be 0. If the response heritability is 0.25, a fairly modest one that's often found, then after 1 generation, the progeny will have moved up 0.5 standard deviations from the mean. If the heritability is 1, then its offspring are going to be right at the same mean as the parents. Evolution takes long periods of time, hundreds, thousands of generations. You can see very readily that a small level of heritability, coupled with even a modest level of selection, can lead to quite remarkable and strong changes after enough generations. Now, in that Thorndike quote that I gave earlier, he used the word plasticity. And I see the future of this type of studying moving more and more to trying to understand plasticity, in which behavioral plasticity, I think, is a key and interesting issue. First of all, plasticity we can define as the fact that different phenotypes can be produced by identical genotypes due to environmental or developmental events. Okay. So the same genotype gets different phenotypes. No problem there. The normal reaction is an interesting concept because it reflects the extent to which the phenotypes resulting from a given genotype are or can be affected by environmental or developmental events. There are various strains of mice, for instance, C57s, DBAs, and many others, that it's been shown respond a little or not at all or a lot to the same kind of variable like handling. Okay. Or aggressive encounters. Some strains have these to long-lasting effects, others to virtually no effect. There's a normal reaction. Now, what we know is that, or we're starting to get information, that there is selection for this normal reaction, for this plasticity itself. So the effect that evolutionary process have on the norm of reaction. This concept, of course, blows the distinction between innate and learned behavior, genetically determined and completely environmentally determined. Okay? They're not that distinct. Even more exciting is the recent work indicating that there can be phenotypic effects feeding back onto the genotype. For instance, the various ways in which environmental and developmental events can affect the activity of genes. We're not talking Lamarckian inheritance of acquired characteristics, but that the experience of the phenotype can turn on or change the expression of the genotype. Now, I'm not going to avoid talking about snakes, giving some snake data. And I want to refer to a study that we've just published on the heritability of responses of baby snakes to chemical cues at birth. Now, these baby snakes, these are garter snakes, they respond to odors of fish or worms, amphibians, rodents, leeches, slugs, whatever, at birth with increased tongue flicking and actual prey attack. So they've never had the prey in their life. Okay. Different individuals respond more or less. But for each species, you get a pretty robust phenomenon that's different. Now, we studied a population of a generalist species of garter snake, Thamnophis hortalis, which in different parts of its range may be eating different kinds of prey. But on Beaver Island up in northern Lake Michigan, they're eating nothing in our population but earthworms. As far as we know, for generations, they've been eating nothing but earthworms. However, they will eat fish. So what we did is we looked at the responses to fish, the heritability responses to fish before and worm before any feeding experience. And surprisingly, we found zero heritability. Other people have done studies like this, and they've always found some heritability, and we found no heritability. Maybe it's because they've been on this island and living on fish, and so any genetic variation was sort of gone. Well, not really, because we fed them all on 12 meals of fish, and then we tested them again. They all had the exact same experience on fish. Now what we found is that after experiencing 12 meals on fish, the heritability for fish went up significantly, 0.32. Furthermore, the response to worm that they never ate, all of a sudden now we had a significant heritability calculated. Lesson 1, heritabilities are not necessarily fixed, even within a population, within an animal. You have to look at different ages. And in fact, even with humans now, there's more and more evidence coming out that some of the genetic effects actually become manifest later and later in life, whereas the general idea was that what you're given with genetically is at birth, and then operation of environment, selection of the consequences, is moving you farther and farther away from your, quote, genetic nature, your genetic heritage. Whereas there seems to be some evidence that maybe experiences tap into and bring out the genetic variability that may be there. If you looked at the preference for fish versus worm, just take a different score. Before any feeding, no heritability. This I found pretty remarkable. The preference for fish, that was highly heritable. The ability to learn to respond to fish. I need to tell you, by the way, that after they start feeding on one prey or another, they can change their preferences. Okay. So the relative preference can change with feeding experience. The amount of change seemed to here to be heritable. Okay, the plasticity of the response was heritable. Not only that, we also looked at how well the animals grew on the diets. Now remember, they all fed exactly the same number of prey, the same number of meals. They all grew well. They were only using animals that survived throughout the entire experiment, which was most of them. And what we have here is that the response to growth, you could predict the growth rates from the response to fish and worm before. However, after they had the feeding experience, the fish response did predict how well they grew on the diets, but not the worm response. And furthermore, what was really interesting is that the preference for fish minus worm afterwards reflected their growth. So in some way, the plasticity of the response, even though they were eating the same amount of food, the direction of that plasticity of the response somehow is tied into their ability to use the prey in growth. So I think that the linkages here between the perception, the learning, the growth, nutritional value, are linked in some very complex ways, and maybe more of that going on. I will finish with sort of what this may all mean. If the goals of behavior analysis are to understand, control, and predict behavior in its fullest, evolutionary and genetic processes cannot be ignored in the study of human and non-human behavior. If the goal is to move populations or groups of individuals to behave differently, such as conserve energy, tolerate differences, reduce aggression, improve education, the selection models of genetics may have heuristic value. Third, the emerging rich understanding of plasticity in genetic and behavioral systems may lead to shifts in the conceptual and methodological approaches of evolutionary, genetic, and behavioral researchers if the contingencies and reinforcers encourage such integration. It has not happened to date. And fourth, behavior analysts need to be familiar with the essential elements of evolutionary dynamics, even if only to be able to critique other evolutionary approaches to behavior. Thank you. Yes.
Loud. Do you have, for the snakes that you work with, do you have an actual gene for the chemoreceptors?
No. However, there is a laboratory in New York, Mimi Halpern's lab, that's getting pretty close to knowing exactly where in the nervous system the chemicals are being picked up, at least for one of the prey types.
Do they have the proteins?
Yeah, that's what they're working on, yes.
So still working backwards to find what the gene.
Right. But there's no question that it's genetically influenced.
Is it known whether you're dealing with a single gene or whether you're dealing with a contiguous gene situation?
I would suspect we're dealing with several genes here. One thing I didn't mention, by the way, is that would complicate this story a little bit, makes it more interesting, is that all these calculations are based on assuming that all the offspring in the litter were fathered by the same dad. Okay. But we actually know, we've demonstrated that there's multiple paternity in this group, and actually we're trying to use that now to assess maternal effects. But
how many chromosomes are
in this particular species, I don't. It's not abnormal, it's typical vertebrate. Yes?
Did you ever look at other populations to see if you had the same correlation between preference for fish and growth rate, or maybe preference for worms and growth rate?
We've studied many different populations and species, not particularly with this design. And we find individual differences in how they grow on different diets. And one thing that we've discovered is that with some species, if you add a little calcium, various things that are in fish and not in worm, then worm eaters will grow faster. And some populations that are adapted to worm, the calcium actually hinders their growth. Yes?
I have a quick, brief comment and one question. I really enjoyed that part of trying to use the formal tools that evolutionary biologists have developed to describe selections and responses to selections, the various types of selections, and try to see if they have heuristic value for understanding behavior and selection at the level of behavior. I'm therefore surprised that you did not include in your review of selection models the one that I find perhaps even more important than all of those, and that is frequency-dependent selection. My question, this was a brief comment, the question is, give me an example of selection for the range of variation. I mean, I'm not familiar. You said that apparently we can increase the normal reaction. I misspoke. You said that we can increase the normal reaction, and there were some examples of that. Could you give me one?
There is a book that came out recently by my colleague at the University of Tennessee, Massimo Pagliucci, that goes into this in great detail. And he works with plants. He's a botanist, but he's got very interested in behavior and psychology as one of the aspects to really look at this issue of Norman reaction. And I'd recommend that book. And the stability, you know, the selection for stability, birth weight in humans is one where there seems to be an optimal weight. If you're too far, too side, there's a selection, less survival of babies.
Last question. A related question, quickly. What reading, single book or article would you recommend to a relatively naive or beginning student in behavior analysis versus a very sophisticated researcher in behavioral analysis who knows a lot of mathematics but not the mathematics of heritability and selection?
I would look at almost any population biology text. The one that I like is one by J.F. Crow, and I may actually have a copy of it here. It's been out for a while. Basic Concepts in Population, Quantitative, and Evolutionary Genetics by James F. Crow. It's a Freeman book. I don't know if it's still in press. That was 86. But there are many textbooks. I would say if you look at almost any evolutionary biology textbook, any textbook that your colleagues are using in teaching evolution, you will find references to this kind of material. If they don't discuss it in great detail, they'll have references to the latest books.